So welcome back, session 3, right after lunch. I hope you enjoyed it. So we're gonna get started with, as I said, the panel 3. I'm very excited, it's very interesting. The state of HIV vaccine efforts and antibody-mediated treatment. How much longer until we have an effective HIV vaccine? And obviously we're expecting an exact date, preferentially, at the end of that session. And we have 3 amazing speakers today, and we start off with Katie Stevenson, who is a physician scientist at the Beth Israel Diakonus Medical Center here, and has extensive experience with vector-based vaccine approaches, and this is what she's going to talk to us about. Thank you very much.
Good afternoon, and thank you to the organizers for inviting me today to speak about adenoviruses and in particular Ad26-based vaccine development. So the rationale behind developing HIV vaccines using Ad26 as its foundation starts with the idea that it's good to use a vaccine vector for which there's not a lot of pre-existing immunity. So one of the advantages of Ad26 is that pre-existing Ad26 neutralizing antibodies are relatively rare in humans. This is important because if you have high levels of neutralizing antibodies to your vector, then you're likely to have decreased immunogenicity of your vaccine. In addition, adenoviruses in general are very good vectors because they can accommodate the HIV inserts, the envelope, the gag, the pol genes that you put in there. And just as a schematic, I put on the bottom of this slide what the Ad26 vector looks like. To make the vector, you start with the wild type, you pop out the E1 gene and replace it with your transgene cassette. This has 2 functions. One is that it allows you to put your payload in, which is your protein, and then it also makes your vector replication incompetent in this example. Ad26 is also biologically and immunologically distinct from other classically described adenoviruses like Ad5. This makes it hopeful that perhaps this unique immune profile will lead to better results in regard to HIV protection. And finally, prototype Ad26-based HIV vaccines have already been shown to be well-tolerated and immunogenic in humans, and this is work done by our colleagues at Brigham and Women's Hospital. So to test whether or not Ad26-based vaccines can actually lead to protection in an animal model, Dan Baruch and others at Beth Israel conducted a study in monkeys. So this study was published about a year ago. There were 32 monkeys in this study. 12 of them received Ad26 as a prime, and then as a boost they received an SIV GP140. GP140, if you recall, is the envelope protein. In this case, this is an SIV model, so simian immunodeficiency virus model. The vaccines express SIV proteins, and the monkeys are challenged with SIV. 12 monkeys, so to recap, 12 monkeys got adenovirus and then followed by GP140. 12 monkeys then got adenovirus and then boosted with a vector, another vector, Ad35, and then 8 monkeys got placebo. All the monkeys that got vaccine, those 2 groups, we did observe a per exposure risk reduction, meaning that in this challenge model, they're challenged 6 times with low-dose intrarectal virus, and then we count up how many times it takes before they're infected. So the vaccines did great in terms of the per exposure risk reduction, but what was really interesting and what jumped out in the blue line is that the monkeys that received adenovirus 26 followed by the soluble GP140 actually had 50% complete protection. So that means that 6 out of 12 monkeys at the end of the challenge series remained uninfected. The immune correlates analysis in this study showed that the driver or the strongest association with protection and number of challenges was the magnitude of binding antibodies, so a very simple ELISA. But it was also the functionality of the antibodies that seemed to matter. This is some analysis that Galit Alter did, looking at the polyfunctionality of these antibodies. And here I'm not referring to neutralizing function, but rather all those non-neutralizing functions that the antibodies have through their Fc portion. So now moving into some unpublished data, this is some work that the next phase of this is, so now we've looked at these vaccines expressing SIV proteins in an SIV challenge. Now to bring it a little bit closer to humans, we want to test the actual HIV vaccines that we're going to be testing in humans. So to do that, you put them into monkeys and then you challenge them with SHIV, which is simian human immunodeficiency virus, HIV on the outside, SIV on the inside. So in this study, there was a very large study. It was also in collaboration with Janssen, which is our industry partner for pulling these vaccines forward, 72 monkeys. And all of these monkeys, except the placebo group, got primed with Ad26. And then they were boosted with a number of different varieties of boosts. And what's important for you to know, I think, about the way this study was structured is that the boosts really break out into whether they're boosted with vector alone or whether they get the GP140 somewhere in that boost. And that turns out to be very important. So all of these monkeys were vaccinated and then challenged with a SHIV virus, and they were given that 6 repetitive low-dose rectal challenge I told you about. So Frank Wegman presented this data in Chicago. He's a representative from Janssen. And the first thing to point out is that the vaccines were immunogenic. So but what we observed right away was that what seemed to be critically important is whether or not that GP140 was in that boost. So priming with a vector, a viral vector vaccine, and boosting with a protein seems to be important, which should not be a shocker if you follow the results of, say, RV144, which had a similar premise. So just to show you on the left, to kind of point out what I mean, the blue group and the purple group, those are groups that got vector only and no protein boost, versus red, green, and yellow. And clearly the red, green, and yellow is higher than the others. So the magnitude of the binding to, in fact, a lot of different GP140s from different clades was higher in the groups that were boosted with the GP140. So functionality is something we're interested in. As I mentioned, that was a correlate in the previous study. And again, it looks like the GP140 groups were higher in regard to the functionality of their antibodies. So on the left is the best example in the vector only group in blue. It's not that much better than the sham group in regard to phagocytic activity. However, the groups that got that GP140, the red, the green, the yellow, those groups were much higher. So then I also did an analysis I presented in Chicago looking at the binding of antibodies to linear peptides. And in particular, I was interested in whether there was more binding to the variable regions in the groups that got GP140 versus the ones that didn't. So in the animals that did not get GP140, I found that it was, they had almost no detectable binding to V2 peptides at all. And it was only the animals that got the GP140 boost that bound, in some cases, up to 50 or 100 different V2 peptides. And this distinction was observed for V3, V5, and then also a subgroup of V2 peptides that include amino acid position 169. This is a motif that's found, was identified in the CIV analysis after RV144. All of these peptides on the particular analysis that we used are many, many different variants representing sequence diversity. But if you align all of those and you kind of try to look for any double counting and you really identify the absolutely only unique epitopes that these antibodies are binding, you still see that the animals that were boosted with GP140 seem to have more diverse V2 specific binding as well, at least by linear peptide analysis. But the big question is, did they protect? And they did protect. So the animals that got the adenovirus 26 prime and were boosted with some regimen that included GP140 all had very good per exposure risk reduction after that 6 challenge series, about 90%. But what was really notable was the green line, which I've shown, which is that Ad26 prime, and then the boost is Ad26 and GP140 put together. In that group, there was 66% protection after the 6 challenges. And the immune correlates analysis for this study is ongoing. It's very comprehensive. And hopefully that data can also be presented. So you may be wondering, are these vaccines in humans? And they are. So the first in-human trial of an Ad26 prime with this soluble GP140 boost, basically the same vaccines, has gone into humans starting about a year ago. This is a trial called IPC-AVD-009. IPC-AVD is the Integrated Preclinical Clinical AIDS Vaccine Development Program, and that's a NIH grant that has been helping bring these vaccines all the way from preclinical to clinical. In this study, by design, it looks a lot like the monkey study. And that's because we want to use the data we generate in monkeys to help us predict results in the humans, and vice versa. This study is actually about 400 people in this study. It's sponsored by Janssen, but it's actually funded by a number of different partners. And the status is that the vaccinations are actually complete. All of the people have received the primary vaccine series, and the primary analysis is also completed. So we are hoping in the next maybe 3 or 4 months that we can start sharing data from this as well. You may notice, just to go back for a moment, these vaccine regimens I'm describing are quite complex. And so it really doesn't, one thing that has kind of been something we've been mulling over is that it kind of doesn't matter how awesome your vaccine is if it's too complex to deliver in a real world setting. So we opened a study at a new clinical trial site that we have at Beth Israel so that we can focus in on whether or not we can simplify and shorten this vaccine regimen. You know, thinking forward in terms of real life use of these vaccines. So we opened a study last spring and we completed enrollment over this summer. There are 36 subjects. And I just want to draw your eye maybe to group 2 to show by example what a simplified regimen looks like, where potentially you could deliver AD26 and GB140 together for a 3 vaccine series that is only 6 months long. So the status of that, as I said, the enrollment is complete, and all of the participants have reached week 12, and we should have data from that in the spring. So I hope I'm making you excited about these vaccines, and part of the reason I'm excited about them is that there's a pathway to licensure for them. So we have an industry partner, we have great preclinical data, and we have first-in-human data on these products. So what's the pathway? So first there's next-generation constructs. So unfortunately, I didn't have the time today to talk at all about the inserts, the design of what you actually put into these vaccines, but there are always tweaks and ways to improve them so that we can increase the diversity of the immune responses that these vaccines generate. And those are in studies that are also already in humans. But really the money is in launching an efficacy study. So to do that, we need to develop go, no-go criteria. So that's criteria that all of our partners can agree on are the immune parameters that are going to allow us to pick what's the best regimen for going forward. And clearly, some of that is going to come from the monkey correlates analysis I mentioned. If it is a go, and we do decide, then the plan is to launch a 2,600-person efficacy study. This would be in South Africa. It would be in partnership with HVTN. And ideally, this would start within a year. So let me acknowledge my mentor, Dan Baruch, as well as Ray Dolan, Lindsay Baden, and Bruce Walker, mentors of mine for many years now, our partners at Janssen, MHRP, and also the members of our clinical trials unit that we just opened this year and of which I'm very proud. Thank you, and I'll take questions.
Thank you very much.
Katie, 2 quick questions. Does the AD26 itself have an ENV insert that you're priming with?
Yes, it does. The AD26 expresses an envelope protein, a GAG protein, and a PAL protein. There's actually 2 versions of GAG and 2 versions of PAL.
Okay, and then is the GP120 boost in an adjuvant, and do you think the AD26 is acting as an adjuvant for the GP140 protein?
Yes, so it's a GP140 protein, and it is delivered with an alum adjuvant, but if we notice that the group that gets GP140 alone as a boost is actually not the best group. The best group is the one with AD26 plus GP140, and we theorize that the AD26 is providing some adjuvanting, maybe because of the TLR agonist that's on the surface of the virus.
So I guess as a follow-up to that question, what do you think is going on with the protein boost? What is the protein boost doing? Is it maybe stimulating more TFH? Because it's a soluble antigen, right? So it's
Right.
So maybe it can target more the endocytic pathway. So maybe that's stimulating more TFH.
We
cells?
don't know, but we're actually going to try to answer that question. We just got an amendment to our protocol with the Reagan support, which is supporting this study, to do fine needle aspirates of lymph nodes. So we're going to do that on our participants who get protein or don't get protein and try to look at some of the TFH responses and see if we can see differences.
Katie, I have a question also, which is over here, which is what do you think, what does your team consider to be the threshold level of protection that's going to be required to make this something that's deployable?
That's seen in an efficacy study, you mean? I mean, I think a lot of that has to do with computer modeling in terms of what is beneficial for different communities. I think that it kind of depends on what your prevalence of disease is and what you would go for. But I think people use like maybe 50% is something that they're looking for. Certainly we know that the 31% that was observed in RV144 was not enough to generate interest in licensing a vaccine. So I think 50 might be something we're aiming for.
Great. Thanks very much. So it's a real pleasure to introduce our next speaker, Johannes Scheid. About 7 or 8 years ago, Michelle Nussensweig and I were talking at an HHMI meeting about opportunities in the HIV field, and we're talking about these elite controllers that we had in our cohort and whether they would have neutralizing antibodies. And Michelle said that he had actually just recruited somebody to work in his lab who was interested in exploring that question. That person was Johannes Scheid, who went on to identify from one of those elite controllers an antibody 3BNC117, which I think is familiar to many people in the audience. Johannes is now a resident in medicine at Mass General Hospital, but continuing to be involved in his research related to the use of broadly neutralizing antibodies as therapy. So, Johannes.
Thanks so much, Bruce. Yeah, let me say it's a real honor and a pleasure, really, to be here today and to talk to you a little bit about our work on the antibody response against HIV. So, as all of you know, one of the big challenges in HIV vaccine design is the incredible diversity of HIV. And one hopeful piece of information that we've known for a long, long time is actually that some patients do develop really potent neutralizing serum responses against HIV. We know that about 5 to 10% of HIV positive individuals eventually mount such a response. And not only that, but we also know from studies in the 90s, transfer experiments, that if you take those sera and transfer them over to macaques, that these antibodies can be protective. So we got really excited about that, this finding, and then started to look at different ways to really get to the bottom of what this activity looks like on a monoclonal level. And so we developed some tools to actually identify and characterize this B-cell response from those 5 to 10% of patients on a monoclonal level. And as a result of that, we're able to isolate a whole group of different broadly neutralizing antibodies targeting different sites on the HIV envelope. And we're joined in our efforts by other groups such as John Muscola, of course, and Dennis Burton. And then one of the postdocs in our lab, actually Florian Klein, then came up with the idea of testing those antibodies, some of those antibodies in vivo models, and he started with humanized mice and was really able to show that those antibodies show effects in vivo models. And again, our efforts were really joined by other groups such as Dan Baruch, of course, and Malcolm Martin, who did extensive experiments in macaques and basically were able to show that not only are these antibodies able to suppress viral load once it's established in macaques, but also can be protective against SHIV challenge. Now, another benefit of these studies was that we were able to really look at some of these different antibodies and figure out which ones would be the most promising candidates to move ahead and do human trials with. And one of those antibodies we're going to be talking about today is 3bNC117. And one of our postdocs actually in the lab, Marina Kasky, was the first to go ahead and test 3bNC117 in a human trial, which was published last year. And this is just one figure out of that study, but basically summarizing the effect of 3bNC117 on the viral load in viremic individuals. These are 8 individuals that were enrolled in that trial. And what you can see is that their viral loads dropped between 1.5 and 2.5 logs after one injection of the antibody, and this effect lasted for a few weeks, at least as long as the antibody was present in sufficient levels. So we were obviously very excited about this and would have been delighted to go ahead and convince the FDA to do a combination trial, but of course had to first proceed with other single antibody trials to really show that this is safe. So this is the trial that I'm going to talk about today a little bit. It's a treatment interruption trial with a single antibody, with 3bNC117. In this trial, we started off with recruiting patients who are on ART, have fully suppressed viral loads for at least 12 months. And we gave the first group in this trial, we gave an infusion of 3bNC117 at a dose of 30 mg per kg, and then interrupted their ART, and then gave a second dose of 3bNC117 exactly 3 weeks later. And obviously followed these patients with their viral loads and other lab values in this time of treatment interruption. A second group we enrolled in a fairly similar regimen, however, with more infusions. So again, these patients stopped their ART after a first infusion of 3bNC117, and then we followed with a second, third, and fourth infusion 2 weeks apart. And during that time, of course, they interrupted their ART. Now, before we enrolled all of those patients, we pre-screened them somewhat with the following assay. So we took between 4 and 8 million CD4 T cells and did outgrowth assays with those cells. We know from studies from Bob Silicano that roughly one in a million of CD4 T cells really carries reproduction and replication efficient virus. And so, we can assume that about maybe 4 to 10 T cells were in those samples that really had replication competent virus. And so we outgrew those samples and then screened them for their sensitivity to 3bNC117. That data is summarized down here for IC50 and IC80 values. And we, in our trial, just used as a cutoff 2 microgram per mil for an IC50 value to enroll the patients. So they had to be able to be neutralized at 2 microgram per mil or less IC50 in order to be part of our trial. And you can see this, that it's about 60 to 65% of patients who met that criteria for 3bNC117 and actually far less for VRC01, which performed less potent in these assays. Then we obviously, as I mentioned earlier, followed the viral loads and also the antibody levels in those patients. And this is the data for the 6 individuals that were enrolled in Group A. And you can see on the left the viral load, on the right the levels of 3bNC117, which are summarized in the red curves, whereas the viral loads are shown in black. And you can see that at least 2 out of those 6 patients were suppressed for at least 9 weeks after stopping ART. And actually all of them had some level of delay, which you can see by comparing their rebound to an average rebound that we derived from a large cohort of ACTG patients who had undergone ATI in previous studies. When we looked in our second group, Group B, again with 4 infusions, the results were actually even much more convincing. We had 5 individuals, 710, 712, 14, 15, and 16, who were suppressed for at least 9 weeks after stopping ART. And again, all individuals except actually 711 showed a delay in rebound compared to the data that we'd seen in so to speak, negative controls from ACTG data. This is another way of summarizing these results. So we have a Kaplan-Meier curve summarizing those ACTG patients that we used as a comparison, and then the combined individuals from our trial. When we looked at so we wanted to figure out in how far this assay that we did in order to pre-screen patients really helped us to predict the effectiveness of 3BNC117 in delaying viral rebound. And so we compared the IC80 of those outgrowth cultures that I described to you earlier with the delay of rebound, and found that there was no correlation whatsoever in Group A or Group B between the IC80 before ATI and the delay of rebound, which in a way makes sense because, as I pointed out earlier, we would expect to have something between 5 to 10 different infected CD4 T cells that grow out in those cultures, and actually we were able to confirm that by sequencing the cultures. So it's a very, very small sampling of what is going on in those patients' reservoir, and therefore really a very random way of pre-screening patients, but we thought it made sense to at least exclude patients who have very pronounced resistance. We also looked at, obviously, the sensitivity of the rebounding virus to 3BNC117 that's summarized in this slide here. And just in the interest of time, I'm going to skip ahead here and just and summarize that basically what we found is that in most of the individuals, so this part down here, this graph down here shows you the difference in sensitivity of outgrowth cultures from the baseline to the rebound samples, and showed that in most of the patients we had increased resistance to 3BNC117 at rebound compared to screen, with some exceptions over here, as you can see, where there was no change in sensitivity to 3BNC117. And on the other hand, we also checked all of those samples against a different antibody, 10-1074, and found that only in one case was there a change in sensitivity to that antibody. We also did single genome analysis of the plasma rebound samples, which was actually quite surprising. These experiments had been done earlier at with patients who just interrupted ART, and it was found that actually rebound in the setting of ATI is usually polyclonal, with many dormantly infected T cells reactivating. And we found, in contrast to that, that the majority of patients that we looked at had a monoclonal rebound. Finally, we also sequenced those plasma rebound samples and looked at escape mutations. And again, we were quite surprised to find that only in 2 individuals, 702 and 703, we found very classic 3BNC117 CD4 binding site knockout mutations. Most of the other patients had mutations in the rebound compared to baseline that were in the area of the CD4 binding site, but were not classic escape mutations. And if you look at the sensitivity curves of TZMBL assays done with those monoclonal viruses against 3B&C117, you will see that, for example, in 709, the rebound is not, which is shown here in red, is really not a complete knockout, but really a shift of the neutralization curve. So suggesting that you really don't need complete knockout resistance in order to rebound. So in summary of what I've shown you, we can say that 3B&C117 administration is, in the setting of ATI, is safe. We know that a large group of participants were suppressed for at least 9 weeks after interrupting their ART. We saw that the longest suppression after 4 infusions of 3B&C117 was in one individual and lasted 19 weeks. The rebound did not correlate with the IC80 sensitivity of the outgrowth cultures to 3B&C117. The rebound was associated with decreased sensitivity to 3B&C117 in outgrowth cultures of the rebound viruses. And we found that most of the viral rebound was monoclonal, which is very exciting because it does suggest that there was significant selective pressure exerted by 3B&C117, and viral rebound rarely involved classic escape mutations. So where is this all going? We're obviously really excited now that 10-1074, a second antibody that's targeting a different epitope on the HIV envelope, is well underway in phase 1 trials. And actually, our group is starting a combination trial fairly soon. And we know actually from animal studies that, if anything, this a combination of several antibodies will be even much more promising in the setting of an ATI, but also of a treatment of viremic individuals. With that, I would like to close and thank many supporters and collaborators. Of course, Bruce, who's been just extremely supportive of all of our efforts from really the first moment on. David Ho, who also helped us with patient samples. The Rockefeller University Celldex Therapeutics, who's making the antibody. The Reagan Institute with Boris, who's helped in that last study. And Weill Cornell, Caltech with Pamela Bjorkman and Mike Seaman here in Harvard, who's been extremely helpful, as well as Beatrice at UPenn.
Okay, people are lining up. Ready? Raj.
Raj Gandhi from Mass General, Harvard, CFAR. Very nice work, Johannes. Are you viewing this strategy as something that might replace ART in terms of long-acting antiviral therapy, or do you have any evidence that this might affect virus reservoirs? Would this be a curative strategy, or really just a long-acting ARV for replacement of current meds?
So I think that's a fantastic question. We don't know the answer to that. We also, one thing that I find exciting, and I'm not sure if other groups are looking into that as a possible clinical trial, is even combining some ART medications with antibodies. What we can say so far is that this is an extremely powerful new arm, a potential new arm for HIV therapy. I think what was really exciting as an immunologist was to see the work last year published from our group showing that actually in addition to suppressing viral loads similar to ART, antibodies also increase, at least in some cases, the endogenous B cell response and antibody response in patients who are receiving the antibody, suggesting that there is some kind of vaccinal effect, if you wish. And I think, in that setting, we still have to figure out what really the role of antibodies are.
Yusuf Urkel from Kernel Biologics. Is the microphone working? Can you hear me?
Start over a little closer. I think they just turned it on.
Yusuf Urkel from Kernel Biologics. First of all, thank you for this very exciting data. I think there's great potential of combination with vaccines that we are currently interested in. I was wondering if you could do dwell into some of the deep sequencing of your patient samples to maybe possibly comment about presence of the rare variants in GP120 mutations that could be making patients resistant to your antibody. You have shown that there is viral escape, but is it possible that some of these mutations were already present as rare variants within these patients?
I think that is a fantastic question, and I think, yes, that is very likely, actually, and that in a way, time to rebound is a measure of the frequency of such escape variants to 3B/NC117.
Just as a follow-up, is it possible to pre-screen patients based on their load of these resistant mutations? Have you entertained that idea for future?
You know, again, we would love to. The issue that we ran into, and that's also why we were so limited in pre-screening patients, is that if someone is on ART, the frequency of their really replication competent CD4 T cells in terms of HIV-infected replication competent CD4 T cells is so low that it's hard to do. We're actually looking into this now and taking some of the trial participants and doing leukapheresis on those, but still, it's, as I said, from Bob Silicano, we're pretty much expecting 1 in a million of CD4 T cells to harbor such virus, and you have to do an outgrowth, because if you just do PCRs on a T cell DNA, what you will get is a mixed bag of replication competent and junk.
So, yeah, we have 2 quick questions, if possible.
Debbie Prasad, Johns Hopkins. As just to follow up on those questions, I think it would be important to have some baseline measure of cell-associated infection. So in your participants, did you look generally at, say, cell-associated RNA expression and whether it changed before and after the infusion? Because distinguishing between delay in rebound versus delayed detection of viremia might be 2 different phenomena in the context of a neutralizing antibody.
Yeah, I think that would be a great idea to do that and to see in how far, T cells that do not, are infected but don't have replication competent virus, and how far those are cleared by the antibody. Yeah.
Ron Swanstrom, Chapel Hill. People vary widely in the reservoir. Do you know if the person in particular who had a 19-week delay till rebound had an especially small reservoir at the beginning?
We would love to know if we had a good way to measure the reservoir. I think that's, that's still...
So an outgrowth assay?
Yes, I mean, and again, that's what we kind of did. But, the outgrowth assay, absolutely, it's the closest we can get. And I think it's a great idea to do that. Yeah.
Okay, thank you very much. And I invite the next speaker and the last speaker of that round. Dr. John Steichen comes from us from Scripps, working with Bill Chief. And the title of his talk is going to be Reductionist Vaccine Design Strategies to Induce Broadly Neutralizing Antibodies Against HIV. Thank you very much.
Okay, thank you. So I'm going to be talking about vaccine design to target PGT121-like antibodies. And PGT121 is a broadly neutralizing antibody that targets the N332 glycan supersite on GP120. So this is a structure of PGT122, a related antibody, bound to the BG505 SOSA trimer. And some of the reasons we like PGT121 is because it's broad and it's highly potent. So it's right in here. It's about 65% breadth, and it's one of the most potent antibodies. And it's also been shown to protect against Shiv challenge in macaques, and it can also suppress viremia when delivered after infection. So we think if you could make a vaccine that could elicit this type of antibody, it would have a good chance of protecting in humans. Some of the challenges with this class of antibodies, though, is that it's unknown what the frequency of PGT121-like antibodies are in humans. So we're working really hard to answer that question, but it's still an unknown currently. And like most bnAbs, it has high levels of somatic hypermutation, and it has an insertion and a deletion. So we don't really know if we could target the germline of PGT121, would we be able to boost it to make it into a bnAb with all these unusual features? And another barrier to elicitation of PGT121 is that, as with most broadly neutralizing antibodies, when you revert them to germline, they lack any detectable affinity for either GP120, in this case, the black line here, or to cell surface expressed envelope. But we're a protein engineering lab, so this is the type of problem we're pretty good at solving. And so we thought we could design a version of the trimer that would bind to the germline. And so this shows the epitope of PGT121 on GP120 here. So it binds between these 4 glycans, N137, N332, N301, and N156. And so because this is a glycan epitope, I use mammalian display directed evolution because that gives the proper glycans on the GP120. And initially I had started by screening libraries against the germline antibody, but I didn't find any mutants that bound to the germline. So I had to use this bootstrapping strategy. So I started with a random mutagenesis library, and I selected against this intermediately mutated antibody, which is the germline, but it has 9 mutations from the mature antibody. And so that identified 2 mutations, N133 and N137, that knock out these 2 glycans, and that improved binding to this antibody. And I also screened another structure-guided V1 loop library against a germline +3 antibody, so it's 3 mutations away from the germline, and that pulled out this molecule that we call 5-mut, which had binding affinity for this germline +3 antibody. We still didn't have binding to the germline though, but when we combined the mutations from these 2 molecules, we ended up with this 7-mut that has weak binding affinity for the germline. And then screening another saturation mutagenesis library against the germline got it down to about 1 micromolar. But this version of the germline has the mature CDRH3, so we wanted to go ahead and revert it, the CDRH3, as much as we could. So we made a few different versions of the germline, and then we screened another library, another V1 loop library, and we ended up with this molecule 11-mut-B, which has micromolar binding affinity to most of our germline-reverted antibodies. And so once we had some what we thought were decent priming molecules, we started thinking about how we could boost in order to guide the maturation pathway to become a bnAb. And so in the simplest case scenario, you might boost with the wild type protein. But even if that worked, we think you would still need to boost with some kind of cocktail which represents the diversity within the PGT121 epitope. And so for this, we designed a variable loop cocktail, 5-member cocktail, based on BG505 SOSIP, but it has the variable loops from other strains grafted on. And one of them is BG505. But backing up, we don't think that this boost is likely to work because the epitopes between these 2 molecules are probably too different. And so in our design process, we had made these intermediate immunogens, this 3-mut and 5-mut, that could potentially bridge this gap to make it a smaller epitope changes. And if that doesn't work, we also have a 7-mut, which can make the epitope changes more gradual. And we didn't know initially how big of a jump we would be able to make, but we had all these potential boosting immunogens to test. And Michelle Nussenswag's lab had engineered this PGT121 germline knock-in mouse, which only expresses the germline of PGT121. And so we wanted to know if in this mouse will our germline-targeting prime be able to prime B cells? And if so, will we be able to boost it along with our boosting immunogens to make a bnAb? And so Amelia Escolano in Michelle's lab immunized with our immunogens with our prime and found that we did get epitope-specific responses in these mouse. And so then we needed to start boosting. And so Amelia tested the serum from these mice by ELISA against all of our potential immunogens. And we decided that whichever was the least mutated immunogen that showed some binding by ELISA is what we would boost with. So after going through that protocol, we ended up with this regimen where we primed with 10 mute, boosted with 7 mute, 5 mute, wild type, and then 2 shots of our variable loop cocktail. So it was about a 5-month protocol. And then we tested the serum for neutralization. And we were really excited to see that serum from 5 of the 7 mice showed some cross-clade tier 2 heterologous neutralization. And here's one of the mice, serum from one of the mice that we ran on a large panel. So this is a PGT-121 sensitive virus panel. In the middle column is 3H3L, which is an inferred precursor of PGT-121, which is less mutated and less broad. But we found that our serum was neutralizing a lot of the same viruses as 3H3L. So we were pretty happy that it seemed like it was going down a PGT-121-like pathway. And Amelia isolated the monoclonal antibodies from these mice, and she found that they were pretty highly mutated, so they had up to 30 mutations, and a lot of them were PGT-121-like mutations. And the higher the mutation, the better the neutralization activity was. So that was really exciting. So just to summarize this, so we had designed germline targeting and boosting immunogens using mammalian display directed evolution. We induced modest broadly neutralizing antibodies in this PGT-121 germline mouse. So we think this is a good proof of principle for germline targeting, that it does have some potential to work. And the most important question now is, how can we translate this to an animal with a more complex immune system? Like eventually we like it to work in a human. And related to that, can we identify PGT-121-like precursors that are present in all humans? And we're working on that. We're doing next-gen sequencing of the naive human repertoire. We think we do find PGT-121-like antibodies in all humans. And we're currently trying to engineer our germline targeting prime to bind to those antibodies that we find. And another question is, will we be able to carry out this boosting protocol in the presence of competition with other naive B cells? And to try to reduce this problem, we're doing a lot of resurfacing of our immunogens so we can hopefully block some of the off-target responses. And we're also we want to do a lot of experiments in these PGT-121 germline knock-in mice to try to increase breadth and make it so all mice respond, and try to simplify the protocol in any way that we can. So I just want to acknowledge Bill Schief and the whole Schief lab, especially Dan Culp designed the variable loop cocktail, and Amelia Escalano in the Nussenswag lab did all of the mouse work in the ELISAs, and Devin did the Newt assays. So, thank you.
So while we're taking questions, maybe we can get the panelists to also come up.
Hi, great talk.
Thank you.
I'm curious, so, obviously these mice have knock-in germline antibodies. Do you have any thoughts on what would happen in a human, how high the chances are of actually recruiting these kind of similar germline genes into a germinal center reaction? And do you think it would make sense to, use those actually as a sorting bait and really look what kind of B cells, what kind of germline B cells would bind to these proteins, and kind of see, is there binding? If you just take any person, me, or like a healthy donor who has never been exposed to HIV.
Mm-hmm. Yep, we're actually doing those experiments with Shane Crotty's lab. Colin is trying to sort our immunogen against naive human B cells. But we think our immunogen is going to need more improvement before it's going to be it probably doesn't have high enough affinity right now to target PGT-121-like precursors in a human right now.
For these 2 mice, do you know if there was a glycan component for the epitope they were seeing in neutralization?
For what are you?
Was there a glycan component for the epitope that they were seeing in neutralizing?
Yes, PGT-121. It binds between 4 glycans.
But did you reproduce that glycan requirement in the 2 mice? So when you immunize the mice,
does the antibody itself have
does
glycan?
the mouse antibody have a glycan requirement for its neutralization?
Oh, I don't think so. No. The immunogen does. The virus does, but I don't think the antibody does. Yeah.
Can I ask a question? Can I ask a question?
Yes.
So following up on Ron's question, so you had very specific viruses that came up, and they were the same ones that are very hot for PGT121, and for the precursor, the more mutated version.
Yeah.
So is there something stereotypic about those viruses, that handful of viruses, that could tell you about how you're converging on that neutralization profile that might be a glycan, like Ron said, or something else that's stereotypical that you might want to veer away from maybe to get increased breadth? Have you guys looked at that?
We have, in the Burton lab, has spent a lot of time looking at the viruses that are neutralized, and we haven't found like a pattern that it is. They all have completely different V1 loops. It could have something to do with that. It's hard to say. Yeah.
Do you have any strategies to keep the CDR3 long in any of the antibodies that you induce? Do you plan to combine any such strategies in the future with your own method of guiding the immunogen?
Yeah, I mean, we're trying to target specific CDRH3s, so that's essentially what we're looking for in the human repertoire. CDRH3s that are very similar to PGT121, and then we're trying to design our immunogen that will bind specifically to those CDRH3s.
To keep it long as well, not just be able to bind certain epitopes, but also to keep the interface wide compared to a narrow binder.
Yeah, I think it will. I mean, we are specifically targeting long CDRH3s with our immunogen. Whether we'll also pull out some smaller ones, we'll have to wait and see.
Okay, thank you very much. So we're going to move on to our panel discussion, and maybe I can ask everyone to quickly introduce himself and then tell us how close are we to get an HIV vaccine. No one wants to start that.
Hi, I'm Alan Engelman from the Dana-Farber Cancer Institute and Harvard CFAR. I'm sort of a last-minute replacement for Bill Chief, who couldn't make the trip.
How close are we to the vaccine?
Well, somebody who studies integration, for those who know me, I think we still have a ways to go for a vaccine, and someone who might be coming from a different perspective, though I was pretty impressed with the data that Katie talked about, and I think that seems to be going in an important direction.
Dan?
Dan Baruch from Beth Israel Deaconess Medical Center and the Reagan Institute. And I think we will know in a few years. We can answer your question in a few years, because there's two major vaccines that are headed towards efficacy trials, and if one or both work very well, then it'll set the vaccine field on one track, and if they do not, then it'll set it on a different track.
Galit Alter from the Reagan Institute side of the table, I think my answer would be, I think we're converging on an immune correlate of protection, so I think that we're getting closer and closer as we design more effective vaccines.
Okay.
So I have a question for the panelists. I mean, one of the things that we did not talk about in this session is vectored immunoprophylaxis, as David Baltimore has and Alex Balaz have talked about it. Originally, Phil Johnson had worked in that area as something that's a preventive. It's not exactly a vaccine, but it's, hopefully creating something that a vaccine might induce. Do you want to comment, any of you, about the likelihood of that going forward? And does anybody have any information about what's happened with the PG9 related vector, this AAV vector that has been in people now, I think, for a couple of years, but we haven't heard anything about it? I'm asking first the panelists, so I'll put somebody on the spot. Dan.
So just for those who aren't familiar with it, what Bruce is referring to is using an AAV, an adeno-associated vector that expresses a broadly neutralizing antibody or fragment or a variant to give chronic long-term expression of a broadly neutralizing antibody in vivo that then might be protective against HIV, and in some way is essentially analogous to a vaccine, analogous to a long-acting antiretroviral, but obviously a distinct mechanism. And studies in mice have been quite promising, I think with a caveat that those are immunosuppressed mice that generally don't have an intact immune system and don't develop an antibody response. Studies in primates have been mixed, with some versions showing longer term expression, other versions showing a rather quenched version of the expression level in which an ADA effect, an anti-drug antibody effect, essentially developing an antibody against the bnAb suppresses expression relatively quickly. Whether that's a monkey-specific phenomenon or whether that will also be seen in humans is not known. And I actually have no information about the IAVI study, but I do know that it was started quite a long time ago, so hopefully there'll be data soon. And this is the first phase 1 study of an AAV expressing a bnAb, which in this case was PG9. So that's in humans, and I presume data should be forthcoming, but I don't know when or how.
Okay. Do you want to ask a question, or should I?
No, I can. The crowd can ask questions too, actually, by the way. We don't want to run that by ourselves. John?
So, Mellors again, hoping for a CFAR in my lifetime. No.
This is a good first start.
What does it tell us that only 5 to 10% of chronically infected individuals develop these bnAbs? Are the clones deleted? Is there anti-idiotypic antibody that prevents the cells from emerging? Why is it a rare phenomenon? What insights do people have? And does that raise the bar for actually inducing these in vivo in a person? It tells me that it's certainly not a favored response or a frequent response.
So in a sense, you're asking, is it really just a portion of the population that might even be capable of generating those? Is that
exactly? And okay, I'll stop and see.
So can I make 2 points about that, John? And I'm not really sure where you're going with your question, but I think one point that I'd like to make is that when Johannes talks about the 5-10%, less than 5%, he's talking about these elite neutralizers. And that's a population of individuals who are able to target the virus incredibly widely. There are other individuals, a much larger frequency of individuals, that can make some cross-neutralizing antibody responses, that can see a diversity of viruses from across the world. What level we actually need, I think, is not really known, so that we can, generate a natural immune response that could get there. But the fact that even among individuals who have a progressive course of disease with loss of CD4 T cells and destroyed lymphoid architecture, the fact that they can still make, in that scenario, a broad neutralizing response tells me that there is a path potentially to making a vaccine that could get us at least potentially to 50%. And so, with immunogens like John talked about and others, we might be able to get there. Whether we get to 90 or 100% is not clear. But the other thing that I do want to mention, and I think that you guys should comment on this, how much do you need to get to protection? The other comment that I want to make is that there have been a number of non-human primate studies over the last decade that have shown protective signatures of protection, including the studies that Katie talked about, and we know from other groups around the country and around the world, where they get protection with the presence of no neutralizing antibodies, and that was true for RV144 as well. So I think that while we're in the process of thinking about how we can get to the point of getting a certain level of neutralization, there are signals that tell us that you might be able to get there even without having 90% neutralizing activity.
Let me ask then, the non-neutralizing signatures, are they hypermutation derived or not?
So affinity matters. There's no question. You need affinity to tether yourself, tight enough to the surface of an infected cell or a virus, so you can stick long enough that an innate immune cell can come and kill. So there's no question that there has to be some affinity evolution in there. But the fact that in acute infection we see functional antibodies that can drive very robust clearance of virus, we know that the virus loads decay very rapidly and they must be mopped up. The T cell can only kill the cell. The virus is mopped up through other mechanisms. So I think that tells us is that you can make them very early on with function, even though they're not as affinity matured as you'd see something down the line.
Kent Weinhold, Duke CFAR. Galit, I guess this is following up on that question and directing it at you. As someone who lives with T-cell polyfunctionality on a daily basis, I'm intrigued with the concept of antibody polyfunctionality. And of those components that make up antibody polyfunctionality, if you throw out neutralizing antibodies, what's your take on what's the most important activity, non-neutralizing, that would be desirable in an efficacious vaccine?
That's a tough one, Ken. Should I say ADCC because you pioneered that? Is that the correct answer?
That'll work.
Okay. I don't know what the answer is. I think that people would like to say ADCC because I think that we have assays that can measure ADCC. I think that there are many things antibodies can do, and I think what we're seeing in at least the AD26 animals is that it's not just one function. And it makes, I think, us wonder, when we look at Dan's data and when we look at other monkey data from Bart and others who've seen protection in the absence of neutralization, is the possibility that you need to control the virus in lots of places in the body. You need to control it in the lymph node, you got to control it in the mucosa, we got to control it in the blood. And so different innate immune cells live at different sites, and so having the ability to recruit different types of innate mechanisms may be advantageous. So maybe when we get enough data together, we're going to refine and say it's only ADCC, but I'm not sure that that's clear. I think for cure, it might be ADCC. So I hope that answers and makes you happy.
I hope you're right.
Doug Richmond, UC San Diego. So one thing that's bothered me from the beginning is you don't see the development of any breadth of broadly neutralizing or any neutralizing antibody response to heterologous viruses for at least a year of chronic infection. Most broadly neutralizing antibodies have a great deal of somatic hypermutation. And so how are you going to elicit these types of responses with 2 to 3 injections?
Do you want to take this, Johannes?
I'll
CRISPR.
answer this. No, okay, go ahead.
There's a microphone over there.
Please use the microphone.
Do you want to go there? Maybe this one?
Oh, this one.
I don't know how efficient the virus is at vaccinating in an optimal way that's now being kind of replicated with the sequence of different envelopes. And so yes, the fact that it takes a long time, is discouraging, but at the same time, this is a very extremely inefficient process that goes on in a patient who is infected and exposed to a rising number of different HIV envelopes. And I think the hope in these vaccine studies in mice is to really boil it down to the 5 or 6 envelopes that really matter, and therefore also speed it up, this process. I do want to say one thing, and that kind of relates to the earlier question as well about how unusual it is that people develop broadly neutralizing antibodies. I found it very surprising that it is that number, considering how many somatic hypermutations it takes in some of those antibodies. And again, a lot of those antibodies, VRC01, 3B, and C117, they have 90 plus somatic hypermutations in their heavy chain gene, which is something we never ever see in a normal B cell response. Plus, a lot of them, if you look across patients, come from the same germline, and what are the chances of recruiting exactly that kind of germline B cell into the germinal center reaction and then kicking off that response? So I'm actually surprised and encouraged that it's that many patients, and again, hopeful that if you have a more optimized strategy with different envelopes, that you could actually achieve it.
I could add that I think, Doug, your question it really speaks to the heart of the challenge of developing an HIV vaccine. Because unlike most acute viral infections for which there is ample evidence of natural immunity. For HIV, there isn't. And so for an HIV vaccine, we will have to do better than nature and not simply do as well as nature. And I don't think that has, I don't think there's an answer to your question. I think that it's fundamentally an unanswered question at this point. But the heart of the challenge of HIV vaccine, well, there's many different ones, but one of the key challenges of an HIV vaccine is that simply recapitulating natural immunity is clearly not sufficient.
Right. This is what's concerned me from the beginning about HIV, HCV, syphilis, TB, malaria. If infection doesn't protect, vaccinologists have a challenge. Right?
But can I mean, Doug, aren't you hopeful when you see the CH505 story and you see how rapidly they were able to induce neutralizing antibodies with that one sort of diversification event? So to get to some level of NEUT in 6 months. I mean, that tells you that, there are processes that can take place earlier than 1 year that can allow the immune system to sort of hit that range of viruses. And so if we can understand how that occurs, and theoretically one could accelerate the process. And I think with all the TFH biology and the FDC biology and the germinal center biology that we're sort of developing now because of this problem, that we're going to learn how to cheat the system so we can be better than nature.
I mean, I hope you're right. I want people to develop a vaccine, and I want people to develop a cure. I'm just worried about the challenges. And the fact that you don't see a heterologous response for at least a year, it just makes me a little nervous. So.
Jerry Zach, UCLA. A theoretical question, since I'm not a vaccinologist. What does the panel feel is the efficacy level that will be needed in trials to actually impact the epidemic?
Well, I think that's the question that was started with Katie's talk. I could expound on it a bit, that it depends who you're asking and it depends where you're talking about. So as Katie mentioned already, the mathematical modelers tell us that the efficacy of vaccine that will have a substantial impact is directly proportional to the incidence and prevalence of infection. For example, a vaccine that might have a tremendous public health impact in South Africa probably has a much lower bar than, say, a vaccine for the US or Europe that would have an impact on the Western epidemic.
Yeah, I suppose behavioral changes will have to be taken into account too. If people think they're protected, they may behave differently than if they're worried.
So certainly that's something that needs to be addressed as well. But I think that most models suggest that in high prevalent areas, then a vaccine that's about 50% effective, if it were actually deployed, would have a substantial effect on the epidemic. What would be of interest to either public health authorities, governments, industry partners, that's a completely separate question.
Yeah, thanks.
Raj?
Raj Gandhi from Mass General. A question for Dan around trial design for vaccine trials. So Katie mentioned that in the AD26 experiment that you're doing in South Africa, there's going to be go, no-go criteria. I'm curious what those criteria are. Have you defined them up front? And also she mentioned if it does go forward, a 2,600 person trial might be pursued. If PrEP becomes increasing in frequency in South Africa, does that account for some use of PrEP in the placebo, in the comparative group? So 2 questions. How do you decide on go, no-go? And how is PrEP going to affect the vaccine trial design?
There are a complex series of go, no-go criteria that involve, of course, safety of the vaccine, which we don't expect will be a problem, and then a fairly long panel of immunogenicity criteria that essentially are a merging of the immune parameters that have been shown to be protective in the monkey model, as well as influenced by the correlates of immune protection seen in humans from the RV144 study. And those are obviously being updated and revised very frequently. But there are formal pre-established go, no-go criteria. And of course, the sample size of the large efficacy trials is heavily influenced by what else is happening in the field, such as incidence rates of infection as well as use of other prevention modalities, including PrEP.
Savita?
Yeah, Savita Power, Miami. Coming back to something that Galit mentioned about cheating the system, I was wondering if the panel could comment on these antibodies that are broadly neutralizing, which are less mutated, like the one that Julie Overbauer described in the baby who developed this antibody within 1 year with very few somatic hypermutation and smaller CDR3 length. How do those kinds of broadly neutralizing antibodies really differ from the conventional ones that you guys have been talking about?
How about you, Bruce? I don't think that we can answer that question, Savita. I think that that's a very important question. I think that that's the goal, is to immunize where you don't need to get to 90 mutations, but can get to only a handful and still have sufficient breadth that you can actually attain protection from infection. But I think that we don't know enough. As far as I know, we don't know enough about the diverging functions between those and those that are highly hypermutated, but I think that that's really where the immunization strategies are moving. I don't know if John wants to comment on this in the Sheaf Lab, if you want to comment on some of these less mutated early variants.
Well, it also brings up, I think, another question, which is, have we really saturated the knowledge that we need around naturally occurring neutralizing antibodies? Or are there a whole bunch more out there that might reveal vulnerable sites that we haven't done? I mean, what does the panel think? Should we still be trying to isolate neutralizing monoclonal antibodies? Are we going to get better than what we've gotten with N6 and PGDM 1400?
Do you want to take that one, John, and then I can fill in my opinion?
Yeah, I'll go back to your question. I was just going to say, in Bill's lab, we're putting a lot of effort into trying to identify only the absolute necessary mutations for each of the bnAbs. So we find that a lot of the mutations are actually unnecessary. So yeah, hopefully we don't actually need to get 90 mutations in order to get a bnAb through vaccination.
So that addresses, I think that, and I think that's the same for VRC01 as well, correct? John, is that there are a lot of mutations that are unnecessary, that just occur along the lineage evolution, and they may not be necessary. So understanding how to generate an immunogen that will get you just as far as you need to go without all that unnecessary is really, I think, sort of creating that path to successful immunization. To get to your question, I think, Bruce, I think it's important to understand the landscape of vulnerabilities across the HIV envelope immunogen. And I think that, we pick up on these 5 to 10% of individuals that may be doing it in the same way, and that's how they get to 5% of this elite neutralizing activity. But there are potentially other sites of vulnerability that we're not looking at, because we're not looking at the individuals who are hitting it at 50% or 40%, and maybe they're doing it in a slightly different way, and that may be an attainable goal through vaccination. And I think that that's something that is important to keep in mind, because there may be what is the elite or the top 1% of the population, but what we may be able to cheat may not be to that level. It may be to the 50% mark, and so understanding how to tailor our immunogens to get there may be potentially a path to greater success in germline knock-in mice.
I mean, just to support that, but at the end, if you have one of these super elite broadly neutralizing antibodies with hits all 200 viruses in your testing panel and has extreme potency, that might be a good antibody for passive immunization or for therapeutic interventions or something like that, where you really have only a few antibodies you're going to give. For a vaccine, you might just need many different specificities at a lower potency, and they're still going to do the job. So you don't need to induce maybe like 1 or 2 of these antibodies who can kill everything.
And that's really important, because when we're talking about monoclonal specificities, that's never what happens in an immune response. So following vaccination, following infection, we're making polyvalent responses of millions of different variants that work together. And I think some of the early work by Michelle showed that a lot of the neutralization activity we were seeing in some of our controllers was mediated by polyclonal pools of antibodies that targeted the envelope in different ways. And together, working as swarms, we're able to contain the virus in these neutralizing assays. So I think that it's true. It's that we're looking for those magic single molecules as opposed to looking at the sort of team players that work together to really hit the virus in the most effective way.
Again, Andre Ford from the Penn Center for AIDS Research Board, as well as the National Board. I want to go back to the question, how close are we to the cure? I find that very enticing, but I also find it very misleading, and this is just my opinion. Because when I have...
Sorry to interrupt, but are you saying cure or vaccine?
Cure, I'm sorry.
Oh, well, cure is the next session, I think.
Okay, all right, then I'll bring it up next section.
But we have Alan.
This is vaccine.
But
Okay.
Alan can talk
Well,
to
yeah.
We're equidistant between cure and vaccine.
Well, let me just add, because I have people coming to me talking about, we're close to the cure, we're close to the cure. I go, well, we're close, but we're yet so far. And I think in Pennsylvania and in Philly, I think Temple came out a couple of years ago, and it was on the news that there was a cure. And I had all these people coming to me talking about there's a cure. And I had to go into a heated discussion to talk about there is no cure yet, but we'll talk about it later.
Simon, Washington, D.C. This is almost a philosophical issue. When antiretroviral therapy came out, you saw a little bit of increased risk behavior. And I think King was making the point that PrEP may lead to a little bit more increased risk behavior. If you got a vaccine that was 50% effective, that means 50% of people are not protected, and they may increase risk behavior. So how did the modelers figure that in? It's more of a philosophical question at this point, but something that struck me when you said 50%.
Well, I think that's a very good question. So the question really revolves around the issue of behavioral disinhibition or increasing risk behavior if one incorrectly feels as though one is fully protected. I think some of the increase in STI infections that we saw this morning with PrEP, I think certainly speaks to that as a possibility. Within the context of HIV vaccine trials, including efficacy trials, then that is often looked at. I notice several people who have been more involved in conducting clinical efficacy trials are here now. And Susan, do you want to comment on that? But I believe that there hasn't been evidence in vaccine trials, at least yet, that there is a lot of behavioral disinhibition. But maybe it's because vaccines haven't achieved that degree of efficacy. Yeah, so Susan, do you want to comment on that?
And come
it
to
is
the
something that is
microphone.
being looked at carefully in the context of vaccine trials.
And I guess I would add, as every time you come up with something new that's not an absolute functional cure, which we're not going to have soon, you need to take these types of considerations into account. And I think as time goes on and we have these types of conferences and behavioral science is more intertwined with basic research, I think hopefully the hindsight gets shorter with every new development, and we can shorten the length of time where these disparities might arise.
Dan, you were doing such a fantastic job. I didn't feel like you needed me at all. But I think that it's exactly what you said. We have not seen behavioral disinhibition clearly in any vaccine efficacy trials. It's not clear from the circumcision trials, which are kind of a similar, it's like a surgical vaccine to some extent that's partially effective but not completely effective. We've not really seen evidence that behavioral disinhibition undoes the benefits of that intervention. I think we see more behavioral disinhibition currently with PrEP, but PrEP is so highly effective if you're taking it that it doesn't undo the positive benefits. It does have the unintended consequences that King was talking about before with the potential for increased rates of sexually transmitted infections. But I think it's one of those things that we're going to need multiple interventions altogether. And if we could get ourselves initially to a 50% effective vaccine, we'd be doing the world a wonderful service, I think, because it's not clear to me that that in and of itself would lead to enough behavioral disinhibition that it would undo whatever benefits we saw.
And I can just add one more thing to that. It's actually the opposite, that in the conduct of a clinical efficacy trial for a vaccine, then typically, or what has been seen in the past at least, is HIV incidence rates actually go down in both groups. And that's to some extent, we think related to increasing link to healthcare as well as standard prevention package given to everybody. And so therefore, what is actually seen is a decrease in incidence rates, even in the placebo group. In the trial, yeah.
In the trial, yeah.
So we haven't had a situation of a clinically licensed vaccine that's effective, but those questions I think are highly important and relevant and need to be studied as clinical vaccines move forward.
Okay, thank you very much. We're going to end that panel here. Thanks to the speakers and to the panelists. And as far as I know, we have a 15-minute break.